Let me start off by talking about some of the physics behind vision. So, when you see a color, let's say you're looking at a red shirt, the red shirt is absorbing all the other colors and reflecting just red. And what you're seeing is something known as visible light, which has a specific wavelength, and anything below or above the wavelength will not be seen. So, infrared light has a higher wavelength than visible light, so we won't be able to see that. Our eyes won't pick it up. And X-ray will not be seen by our eyes either because it has a lower wavelength than visible light. So everything you can see is on the visible light spectrum, which is basically all the colors
that exist out there.
Anyways, that's your physics for today. Let's go into anatomy. So let's start off by labeling parts of the eye, and then we'll move on to talking about how the light travels through your eye and how it gets processed by your brain. So without further ado, here's the anatomy of the eye. So on the outer layer, we can find the cornea, which is a protective shield. And then we have the pupil, which is controlled by the iris. So you can think of the pupil as a shutter of a camera. So it is what controls how much light is sent into your eye. If there's too much, it'll contract. If there's too less, it'll dilate. And what's controlling this dilation and contraction are muscles called the iris. Then the light is focused onto the lens, which is right behind the pupil. And the lens focuses the image that the eye is seeing onto the retina. So retina is right here. So one interesting thing that happens that you might already know as a fun fact is that your eye flips the image that you're looking at. So let's say you're looking at a candle. So the image of this candle over here is flipped by the lens, and it becomes something like this. And it's projected onto the retina. So just know that your retina is something like a screen, and between the lens and the retina is something known as the vitreous humor, which is all this empty space in between, and it's basically gel that helps the image get from the lens to the retina, and it also helps keep the shape of your eyeball. If the vitreous humor was not there, then your eyeball would be flat, which would be a problem because the image that you're looking at will not get from your lens to your retina. So on the retina, you'll find two different kinds of cells that are activated by different kinds of lights. So you'll find cones and you'll find rods. Cones are activated by color, and rods are activated by colors on the grayscale, which is basically white and black, and combinations of white and black. So we should also note that there are way more rods than cones. It's about a 10-to-1 ratio of rods to cones in your eye. One thing I want to mention here is that there is a high concentration of cones in a place called the fovea, which is a indentation in the retina. It's not really visible in this image over here, but it's right behind the lens, and that's where there is the highest concentration of cones, and it makes sense that it's right behind the lens because that's where most of the image is going to be shown. And all the rods are located on the sides of your retina, so the extremes, so somewhere over here and somewhere over here. And they are responsible for your peripheral vision, which is normally black and white. So that was the anatomy of the eye. So now let's take a look at how the light passes through your eye. So like I said before, it starts off at the cornea, which is the protective layer of your eye. And then it goes through your pupil, which controls how much light enters your eyes, and the light is focused onto the lens, which then focuses the light onto your retina. And your retina is basically a screen on the back of your head. And there are different photoreceptors on your retina which sense color and black and white. So the photoreceptors that sense colors are called cones, and the photoreceptors that sense black and white are called rods. And once the rods and cones are activated, they activate something known as bipolar cells. And these bipolar cells are neurons, and once these are activated, they activate ganglion cells, which are also neurons. And the axons of ganglion cells make up the optic nerve. The spot where the optic nerve exits your retina, which is right over here, is called the blind spot, because there is no concentration of rods or cones in that area. So now that the information has been transformed into an electrical signal that can travel down your optic nerve into your brain, let's see what happens after the information enters your optic nerve. Once your information enters your optic nerve, it goes straight to the occipital lobe. But this is where things can get kind of confusing. The right side of each retina send information to the right occipital lobe, and information from the left parts of your retinas go to the left occipital lobe. But where it gets more confusing is that the right side of each retina picks up the left visual field, and the left side of each retina picks up the right visual field. So it'll look something like this. And the spot where the optic nerves cross is called the optic chiasm. And the information from your eyes are on their way to the occipital lobe after they cross over and do all that sort of stuff. But now, before they reach the occipital lobe, they have a stop at the thalamus, or more specifically, the lateral genicular nucleus, which is also called the LGN. And I forgot to draw that out in this picture, but it's somewhere over here in the middle of the brain on both sides. And basically, every sense except for smell goes to your thalamus before going to their specific cortices, and this applies to vision as well. So it first stops at the thalamus, and the thalamus, or more specifically, the LGN, decides where the vision is supposed to go, and that would be the occipital lobe and the visual cortex in the occipital lobe. And the image is sent to the occipital lobe where it is processed. So after it reaches your occipital lobe, the image is processed, and there's a famous study done by David Hubel and Torsten Wiesel, who were given a Nobel Prize Award for this study, and they found out that there are called feature detectors in your visual cortex, which basically sense the straight lines, the curvy lines, the different shapes in the image, and put all this together to make one whole image that your brain can interpret. So going back to the example of our candle, the feature detectors are going to sense 4 straight lines, and then a curvy line, and another curvy line, making up the flame, right? And all these are put together, and all the flipping over happens here. Remember that it was originally flipped over in the eyes. Now everything is flipped back to the original form, and your brain perceives this, and that's how you see, basically. So one thing I want to note here is the difference between sensation and perception. So sensation is basically raw data that you input into your brain. There is no understanding or anything like that. It's literally just inputting information into your brain. Perception, however, is the brain's interpretation of your senses. So all these sensation happens in your lens and part of the retina, but once the retina sends the information to your brain starts the perception of the information, which happens, like I said, in the occipital lobe with the feature detectors. And another thing that I also want to mention is a term called transduction, which is where the raw data, which is just the image, is transformed into electrical signals that can be understood by the brain. And if you can recollect back, you'll figure out that it happens in the rods and cones, where the rods and cones basically get activated by the image, and they send info to the bipolar cells, and then the ganglion cells, and then to the thalamus, and then to the brain, and then to the occipital lobe. So that was the basics of vision and how vision is sensed and then perceived by your brain. And overall, I hope you learned something and enjoyed the process. And also, thank you a lot for 250 subscribers. It's a great milestone. So thank you for listening, and this is the Brainy Heart signing off. I'll catch you later in the next video.